I didn't realize that there was like a Stanford Berkeley thing happening in this audience. How many people here from Stanford? How many people from Berkeley? Stanford, I'm disappointed, man. You guys. It's not so much the numbers, it's the enthusiasm that we're missing. Anyways, both fantastic institutions, and I thought what I would do is take the time here to give you the backstory of bioorthogonal chemistry, which as happy as I am at Stanford, it is truly a Berkeley story. So I'm actually going to talk about work that we did at Berkeley between the years roughly 1996 to around like 2010. And that's what I consider the kind of first part of the title here, which was the basic science part. And then I moved to Stanford in 2015, and my lab has really shifted a lot more towards the translation part. So at the end of the talk, I'll just give you some examples of what people are doing right now in the biopharma industry using bioorthogonal and click chemistries as foundational tools. But to kind of give you the origin story, I have to back it up a bit and take myself back into the mind frame that I was in back during my PhD and postdoctoral training, when the idea of what I later termed bioorthogonal chemistry came into my mind as a tool that would enable fundamental studies in the field of glycoscience. So that's another area that my lab has a long-standing interest in, which is the biology of sugar molecules, or glycans, and particularly how they contribute to cell-cell interactions in immunology and in oncology. So when I was a grad student, my focus was in synthesis, in organic synthesis. And how many people here in the audience are organic chemists? Because I'm talking to you today. Yeah. Where's all the cheers, you know, like, go organic chemistry. And for those embarrassed people in the audience who are not organic chemists, let me introduce what we think of as like the fundamental tool of the organic chemist, and it's the round bottom flask. So when I was a grad student, I did a lot of chemical reactions where I combined two reagents or more and to form a product. And the organic chemist is accustomed to doing those kinds of reactions in the round bottom flask using the gadgetry that you see here. And the reason that we have this gadgetry is because this allows the chemist to have full control over all the parameters of their reaction, meaning that the chemist can control the concentration of reagents, the solvent that they're dissolved in, the atmosphere above that solvent, because many of the reactions we like to do in organic chemistry are sensitive to air and moisture. We have to exclude them, so we purge the atmosphere with nitrogen gas or with argon gas. We control the temperature, so we can heat up the reaction if we need it to go faster. We can cool it down if there's some unwanted side reactions going on. And having all of that control allows the organic chemist to perform thousands of reactions that have been developed and described over more than 100 years. But as my own training progressed and I became more and more interested in studying processes in biological systems, I got interested in the idea of doing chemistry not in this well-controlled round bottom flask, but in much less controllable biological settings. For example, I became interested in the idea of doing chemical reactions in living cells for purposes of attaching imaging probes to molecules of interest so that one could study them in living systems. I started thinking about doing chemical reactions in laboratory animals, like the mouse or the zebrafish, to study biological processes in even more authentic settings compared to a plastic dish with cells growing in it. And I started wondering whether there might be a time when one could do chemical reactions in perhaps the most complex vessel one could think about, which would be the human being, for purposes of drug delivery or for diagnostic applications to improve human health, basically. And it was that thought process that ultimately led to this concept of bioorthogonal chemistry, which, if you haven't heard the term before, literally means chemistry that is orthogonal to biology, or put another way, chemical reactions that neither interact with nor interfere with a biological system. And the way that my lab over many years has thought about coming up with these bioorthogonal chemistries is to contemplate the different galaxies of chemistry in the universe of all possible chemical reactions. And it's a pretty vast universe, but there is a defined galaxy within it, which is the biological reactivity space. And that space defines all of the chemical reactivity of molecules, you could say on Earth, or we could just focus on the human body, right? All the different chemicals in your body constitute the biological reactivity space. And it's a large space, but it's not infinite. And elsewhere, whoops, I clicked something. Elsewhere in that universe, oh no, I don't know what I did. There, we thought, could be a bioorthogonal reactivity space, a collection of chemical functional groups that don't exist in nature. So these would be chemistries invented by humans for humans. And they would be chemicals that would be so different from the biological chemicals that there would be no crosstalk between them. And this is the space that my lab set out to discover way back in the mid-1990s when I started my first academic position. Now, it wasn't in a total vacuum that we thought about this concept of bioorthogonal reactivity space. There was a very specific application that I had in mind that drove our lab to focus on this new chemistry. And this, again, takes me to my own interest in glycoscience, which started during my PhD years, but really blossomed when I was a postdoctoral fellow at UCSF. And who here is from UCSF? Cheers. Do we have a mascot at UCSF? There's no animal. I guess when you don't have undergrads, you don't really need a mascot because there's no sports teams or anything. But anyways, I worked in an immunology lab over on Parnassus. This was before the Mission Bay campus was built. And in that lab, I was studying changes in the patterns of cell surface glycans that accompany disease. And at that time, it had already been well established that there are particular phenotypes that are defined by cell surface glycans that are quite stereotypically associated with malignancies. And one of those was the observation that healthy cells have a certain pattern of sugars that terminate with a particular building block, a monosaccharide building block called sialic acid. And so the glycoscientist has a symbolic lexicon that we use to describe our structures. And sialic acids are always shown pictorially as these pink diamonds, and then for those organic chemists, that's what the actual chemical structure looks like. And all of our normal healthy cells have what I like to think of as a well-manicured garden of these sialoglycans, these sugars that terminate with the pink diamond. But the observation was made decades earlier, actually, that if you analyze the sialoglycans on cells in a tumor, cancer cells, you find that they are basically that well-manicured garden has overgrown into like a tropical jungle. And there's an increased density of these sialoglycans on the cancers. And at that time, it was just an observation. It was correlative. No one really knew why. But it's true that whenever you can observe a molecular difference between the disease and the normal tissue, that is a potential opportunity for diagnostic imaging targeting or even therapeutic targeting. And so I became interested in this observation, and I couldn't help but wonder if there was some mechanism by which you could image those sugars, the sialic acids, maybe you could detect and monitor tumors in patients through non-invasive imaging techniques like magnetic resonance imaging or PET scanning, for example. But at that time, and this is now in the kind of early 1990s, there was no technology available for imaging of sugars. By contrast, there was a lot of exciting work going on in the imaging space for other kinds of molecules. At the research scale, people were really excited about genetically encoded fluorescent proteins. So now, you take for granted that you can fuse the green fluorescent protein or M-cherry, right, to your protein of interest and image that protein in cells or animals. But in the 1990s, that was a brand new technology, and everyone was really excited. And those of us who study sugars were really jealous because we didn't have anything like that to study the structures and the changes of these sugars. So I became interested in that problem, and I was in my head thinking about how could you image a sugar in a living system? And science is a funny thing where there's so many breakthroughs that we have intellectually just by virtue of random collisions with people from different areas of science that we might chat with at a conference like this, for example, which causes us to think differently and to go back to our labs and switch entirely the directions of our research in some rare occasions. And I had that experience when I was a postdoc. So my advisor, whose name was Steve Rosen, he was an immunologist, he had agreed to go to a kind of little boutique conference in Southampton, England, to give a short talk on the research in our lab. And this was the conference, it was the Joint Anatomical Societies of Germany and Ireland. They don't even exist anymore, actually, today. But at that time, these two societies would get together and have a conference. And I went to this conference in place of my boss because at the last minute, he regretted he had agreed to go. And he said, who from the lab would like to have a free trip to Southampton, England to give this talk for me? And I was like, pick me, So there I was, and I went to that meeting. And I was a fish out of water in so many different ways. I was the only person there under the age of 65, and I was about 27 at the time. I was the only female speaking at that conference, and I was the only person at the meeting who was not an anatomist talking about organ systems in the body. I was going there to talk about cell surface glycans. And there was one other speaker and participant at that meeting who was almost as out of place as I was, and it was this gentleman, Werner Reuter, a German biochemist, who also had agreed to give a talk at that meeting and regretted that decision. Because like me, he didn't fit in either, but amazingly enough, he was also interested in glycoscience and was studying the biosynthesis of sialic acid. And what Werner's group had discovered, and he talked about at this meeting, was that one could basically introduce chemically modified metabolic precursors of sialic acid into cells, and the cells would transform those precursors into chemically altered sialic acids, which would then appear on the surface of the cell. And so what his group had done was to take this metabolic intermediate called N-acetylminosamine and replace what's normally an acetyl group, so the R would be a methyl group if this is an acetyl group, and he would replace that with extended derivatives where the R group was a little bit longer, like an ethyl group or a propyl group or a butyl group. So one carbon homologs, as we would call them. And as long as that modification was not too large and disruptive, those altered derivatives would get processed in the cell by about 6 different enzymes, converted to the corresponding sialic acid, where now that side chain is right here, and eventually, they would make their way into those cell surface glycans where that modified side chain just came along for the ride. And this was an observation that I don't think anybody in that room cared about. But when I saw this, I couldn't help but wonder, what if you could slip a little chemical reactive group into that modified side chain and then use that chemically reactive group to do a chemical reaction with an imaging probe, okay? And so it just so happened that I heard that talk, and I had a lunch with Werner during that meeting, and we chatted, and I asked him a million questions about what works and what doesn't work and all these things. And then a few months later, I was ready to apply for academic jobs. And many of you are going to do this yourself at some point, and so what you'll find is that when you apply for jobs, you have to write some research proposals that describe the work you plan to do in your future lab. And so I did that, and my sort of main proposal was a 2-step idea for imaging sugars in living systems, which would leverage that principle that Werner Reuter had established. So this is literally the graphic from my job application back in 1994. This was a very sophisticated graphic at the time, and I was really proud that I figured out how to make the lipid membrane and everything, because we didn't have bioart. Okay, you had to do everything using very primitive packages. But the idea was to put a chemical group into a simple sugar, feed it to cells, let the cells metabolize it. The cells will put it in their cell surface glycans, and now it's outside the cell and available to do chemistry with a probe. And the probe would have its own chemical modification, so let's call that group Y, where Y would be complementary to X, and X and Y, if they encountered each other in the living system, would react to form a bond, and now there's an imaging probe on the sugar. And that was going to be my platform technology for imaging cell surface sugars. Now, of course, to do this in a living system, in live cells or in laboratory animals or maybe human patients, those two chemical groups have to be bioorthogonal. They have to not react with other functional groups in the living system. They have to be mutually, selectively reactive with each other. And I had not coined that term yet. We came up with that term a few years later. All I said in my research proposals is that we would choose chemical functional groups that would be very selective for each other. And amazingly enough, at that time, I actually did not know what functional groups could work in this context. And so I didn't even include any ideas for functional groups in my job application proposal, and nobody even asked me. So there I was, defending my proposal in interviews, sweating, like, what if somebody asks me, what are X and Y going to be? And I'll have to say, we're going to figure that out later, Because at that time, you could flip through the thousands of chemical reactions known to the organic chemist. Not one of them really had the qualities of bioorthogonality. So there really wasn't a chemistry that was suitable. But I kind of slipped through the cracks, and I got my job at Berkeley, and I started recruiting students, and the first thing we started to focus on was like, we'd better come up with a reaction, because that's a fatal flaw of this proposal otherwise. So the first reaction we developed, which I think of as really the foundational bioorthogonal reaction, was a modification of a well-known chemistry called the classic Studinger reduction. And this was chemistry that was first reported by a famous chemist, Hermann Staudinger, who in fact won the Nobel Prize in chemistry back in the previous century. Not for this work. He won the Nobel Prize for work in polymer science, actually, which was very impactful. But more than 100 years ago, Staudinger published on this really cool reaction between triphenylphosphine and an azide. Where the azide is this 3 nitrogens linked together. This was a chemistry I was very familiar with because I had performed Staudinger reductions during my graduate school days. And so what happens in this reaction is that the phosphine is a soft nucleophile, and it reacts with the azide as a soft electrophile. And through the expulsion of nitrogen gas, which is a thermodynamic driver of this chemistry, an intermediate is formed, which is called an azaylide. So that's where you have phosphorus and nitrogen bound together with a negative charge on one and a positive charge on the other. So that's the first intermediate that forms in this reaction. Then a person adds water, and water will hydrolyze the PN bond to generate a primary amine and the phosphine oxide. So the amine is the reduction product of the azide, and the phosphine gets oxidized in the process. What I loved about this chemistry, as I had performed it, is it's very selective. You could put your azide into really complicated synthetic molecules and do this reduction with triphenylphosphine without any interference of the other functional groups. It's very mild, very selective. You do this reaction in water, so it proceeds in water, and water is a reagent, in the second step. So in my mind, this was almost bioorthogonal because neither phosphines nor azides are known in nature. These are made by chemists for chemists, and they react with each other and not with other biofunctionality. But the problem here was that although these two reagents, the phosphine and the azide, will form this linked adduct, that adduct is not stable in water. it falls apart, it hydrolyzes. So if there was some way a person could convert the azacylide intermediate into a stable product that doesn't fall apart, that might allow us to do the imaging in biological systems. I should also point out that azides are very small functional groups. There are 3 atoms, but the radius of the azide is on par with a simple methyl group. So I thought we could put the azide into sugars and it wouldn't be a big structural perturbation, right? So all of that led us to develop a just kind of slightly altered version of this classic 100-year-old reaction that we call the Studinger Ligation. And in order to form a stable adduct, we simply introduce this methyl ester on one of the benzene rings, ortho to the phosphorus. And so by doing so, the intermediate azacylide now has a new path of reactivity, where that nitrogen atom can cyclize into the ester, cleave the ester, form an amide bond, and those kinds of intramolecular 5-membered ring-forming reactions are extremely fast. They're so fast that even if you run this reaction in water as the solvent, which of course you would have to do in a biological system, the water does not compete with this cyclization. You get quantitative conversion to the cyclic amide, and now water can mosey along, cleave the phosphorus-nitrogen bond, no harm done there because the amide keeps the 2 components linked together. So this was the first bioorthogonal reaction that we took into living systems to do imaging experiments with sugars. And we already knew from the work of Werner Reuter that getting the azide into sialic acid might be as simple as putting this azidoacetyl group onto that metabolic precursor, which is the minosamine derivative, feeding it to cells, letting the cells metabolize it, and then watching the azidosialic acid appear some hours later in the cell surface glycans. And that worked beautifully. And that became the playbook by which we and others went on to introduce azides into other kinds of sugars. We did a lot of work in those early days putting azides into fucose, which has its own interesting biological story, as well as other sugars like N-azidoacetyl-galactosamine, or GalNAc, which also has some interesting biological stories behind it. And anywhere we put these azides, we could conjugate them with a phosphine reagent via the Studinger ligation. I had 2 fearless grad students who were the first to do this chemistry in live animals. Those folks were Jen Prescher, who's now a professor at UC Irvine, and Danielle Dubey, who's a professor at Bowdoin College. And what they found is that you can inject into mice this azidoacetyl minosamine derivative, let the cells take it up and metabolize it, and then find the azidosialic acids on the cells and tissues of those animals with no harm to the animal that we were ever able to observe. And then if you inject into those same animals probe molecules that have that Studinger ligation motif, you will find the product forming on the cells inside those animals. So that was kind of like a big milestone for the group and really for the chemistry community, because it kind of set the precedent that a chemist should expect that they can do chemistry in a living animal. It just has to be the right kind of bioorthogonal chemistry. But we also learned about some limitations of the Studinger ligation during the course of these experiments and during these years. And it had to do with the kinetics of the reaction. So when you do reactions in round bottom flasks, you can control the rate of your reaction in a number of ways. You can change the concentrations of the reagents, and you can heat up the reaction, right? Those are tools at your disposal. But you don't really have those tools to work with when you're doing the chemistry in living systems, okay? So for people who, I think you probably all took chemistry classes, even though you're not chemists now, which means you know what a second order rate law looks like. Huh? Yeah, sort of. It's all right. I'll remind you that for second order reactions, which is true for most of these like bimolecular reactions where 2 things come together, the rate is equal to an intrinsic rate constant times the concentrations of the 2 reagents. So for us, that would be the azide and the phosphine. And in a flask, you can control these concentrations to make the reaction go faster. But in living systems, you really don't have much control. over these concentrations. For us, we put the azides into cell surface sugars, so there's just an inherent limitation on the density of these sugars as they're displayed on cells. And then for the phosphine reagent, there are some practical limitations regarding how much of that you can inject into an animal without kind of saturating its liver metabolic capacity, and there's solubility limitations and things like that. And also, unlike the round bottom flask, which is a closed system, the animal is not an equilibrium setting. The minute you put reagents into an animal, they're getting cleared out of the system, either through metabolism or just getting filtered through the kidneys and urinated out. So the clock is ticking on your reaction. And if the rate is too slow, the reagents might get cleared before the reaction can occur. And what we discovered with the Studinger chemistry is that as great as it is and as clean as it is, it's kind of sluggish. It has an inherent rate constant around 10 to the minus 3 per molar per second. And what that means on the ground is when we did these experiments in animals, we had to inject the animals every day for multiple days in order to get enough reaction to occur so that we could visualize the product. And that was really too slow for many of the imaging applications that we had in mind. So we decided we needed a faster bioorthogonal reaction and probably at least 2 orders of magnitude faster to get where we wanted to go. So this is around, this is the early 2000s, like 2001, 2002. And we were playing around with some ideas. And I should mention that around that time, there was an uptick in interest around a different reactivity of the azide. And we loved the azide. The azide was just great. So easy to put into sugars, so small, had such interesting reactivity, totally inert in the bodies of animals. And the other thing that azides can do is they can act as 1,3 dipoles and do dipolar cycloadditions with alkynes. And this was a chemistry that was published on extensively back in the previous century by another very famous German chemist named Rolf Huisken from the University of Munich. So it turns out that this other chemistry, which forms a product called a triazole, is, to my mind, almost bioorthogonal. And almost, not quite, because as slow as the Studinger chemistry is, this chemistry is even slower. So when people run these cycloadditions to make triazoles in the laboratory, they have to heat up the reaction, usually to like 100 degrees, in order to accelerate the kinetics, so the reaction will go in a reasonable time frame. And again, you can't do that to cells or animals, right? You can't reflux them in toluene the way that you can do molecules in your fume hood. So we knew that the classic Huisken cycloaddition would be too slow for our needs. Now, in 2002, my Nobel laureate comrades, Morten Meldal from University of Copenhagen, and Barry Sharpless, who collected his second Nobel Prize from Scripps, published just within like 2 months of each other on a mechanism to accelerate that dipolar cycloaddition through the use of a copper-1 catalyst. And what is now synonymous, really, with this term click chemistry, which I learned in Stockholm, was coined by Barry's wife. She's a writer, and she's really good with like the written word, right? And so he was describing the attributes of a chemistry where 2 things come together and very cleanly form a product in very high yield. And she said, oh, it's almost like they click together, and that became click chemistry. And one thing I've learned is even if you're not a chemist, especially if you're a biologist, you might have heard of click chemistry. So how many non-chemists have heard of click chemistry? And for how many of you is that the only chemistry you know of? Probably, yeah, it's not surprising. So this reaction where you combine, in this situation, a terminal alkyne with an azide to form the triazole with a copper catalyst, this is now one of the most important chemistries in certainly chemical biology. It's extremely fast. So this chemistry is done in under an hour at room temperature. You can do it in water. You can do it on complex biological molecules. So people, including us, use click chemistry to conjugate things to proteins and so on. Incredibly powerful. But we also knew that this wouldn't solve our needs either. And the reason is that that copper catalyst is toxic to cells and to animals. So as great as this reaction is as a tool in the chemistry laboratory, it's not that useful if you need your system to be viable. Okay. So we were thinking along kind of a different trajectory about how you might accelerate the azide alkyne cycloaddition, but without using a toxic metal catalyst. And I teach organic chemistry. I've taught that class really pretty much my entire career. And by that I mean sophomore, pre-med, organic chemistry, right? And at Berkeley, that class was Chem 3. How many of you TA'd Chem 3? Nobody. Took Chem 3? Took Chem 3 with me, maybe even, since I was there for 20 years teaching Chem 3. No? All right. At Stanford, we call it now Chem 35. How many people TA'd Chem 35? Took Chem 35. Yay, a few people. So you suffered through either me or someone else teaching you about ring strain. And this is a standard set of lectures that everybody hears when they take this class. And at Berkeley, we would teach this class from either Volhart or Streitweiser and Hethcock, both faculty at Berkeley. And all of these books have a chapter where we describe what happens when you take a very comfortable ring, such as a 6-membered ring, which people think of as essentially an unstrained ring. And it's unstrained because the bond angles in cyclohexane are almost exactly what an sp3 hybridized carbon atom would like. And students, what's the preferred bond angle for sp3 hybridized carbon? It's tetrahedral, so you don't have to really be a chemist. 109. Pretty good, 109. Okay. And you're pretty close to the ideal bond angles in a 6-membered ring in the chair conformation, right? Memories are coming back. But as you compress the ring down and you force those bond angles to be far away from 109 degrees, for example, 60 degrees, like in this little cyclopropane, that uncomfortable bond angle creates strain in the system. And very strained rings like to react so that they can release the strain. And this is really clear when you look at what happens when you put double bonds into these small rings. So double bonds, these carbon atoms are sp2 hybridized. Do you remember the preferred bond angle for sp2? 120 degrees, right? So now you're forcing these bond angles very far away from 120, and that's a lot of strain. And so the double bonds in these very small ring systems are really reactive. They want to do anything they can to get out of the sp2 hybridization, even if they can only get to sp3. And that's a well-known phenomenon. So there I was, writing that lecture to give, like I always do to my students, and when you're teaching, you often have thoughts that can help in your research as well. And the thought that came to mind was, has anybody ever looked at the reactivity of a strained alkyne with an azide? And so I talked to my students about this, and I said, go to the library. This was before the internet. Okay, so I said, go to the library and see if you can find any example of this in the literature. And sure enough, one of my students found this incredible paper published in German back in 1961 by another famous chemist, German chemist, named Georg Vidig, who also won the Nobel Prize in Chemistry in the previous century. And oh, by the way, Vidig was a visiting scientist in Staudinger's lab before he became famous in his own right. So it's like 6 degrees of some kind of German chemistry separation or something that I am now a part of, apparently. But what was published in this paper, they were looking at the reactivity of different kinds of alkynes with azides to react to form triazoles. And they made the observation that this crazy-looking molecule, where the alkyne is now constrained in an 8-membered ring, that's called cyclooctine, is very reactive compared to linear alkynes, okay, which is the more standard alkynes that Hughes had studied. There is almost 20 kcals per mole of strain in this ring. You can't go any smaller with an alkyne in the ring if you want it to be stable at room temp. So cycloheptine, the 7-membered ring alkyne, That was known, but had only been studied at very low temperatures and wasn't stable at room temp. So this is pretty much as strained as you can get and have a molecule you can work with. And this was a German paper, and we didn't speak German, and I had no German postdocs, which was an oversight at the time. But you don't have to speak German to understand what happened here. You can read right here that when they combined phenylazide, which is this, with cyclooctane, which is this, they got an explosion. And we're like, wow, that's promising. But then when we dug a little deeper, we were comforted by the realization that in this paper, what they had done was combine neat cyclooctane and neat phenylazide. They're both liquids. They just mixed them together, no solvent, no dilution. So boom, went off like a rocket. But it turns out if you dilute them down to concentrations that a person would think about, using in a biological setting, you wouldn't expect to have an explosion, okay? So we thought this might be a precedent for a so-called copper-free form of click chemistry, where we used ring strain as the accelerant rather than a copper catalyst. So I had a sequence of students and postdocs who worked on this over many years, and I don't have time to talk about all their work, but I'll show you all of their faces here. Nick Agard was the student who found this paper and kind of launched this whole project in my lab. Then he passed the torch to Jeremy Baskin, who's now a professor at Cornell, and Scott Laughlin worked with Jeremy. He's now a professor at Stony Brook. Ellen Sletton contributed to this. She's a professor at UCLA, John Jewett at Arizona, Gabby De Almeida, who's a consultant at BCG. And over many years, they made all kinds of derivatives of cyclooctanes, decorated with all kinds of ornaments as we figured out how to tune the reactivities. And these are some of the molecules we made. And I'm just going to pull out 4 examples to illustrate some trends that we learned in this process. So up here in the upper left is one of our early derivatives. It's kind of vanilla, in its ornamentation. And we measured the second order rate constant of its reactivity with azides, and it clocked in at around 1 × 10⁻³ per molar per second, which you might remember is about on par with the Studinger ligation. So that was where we started, and we knew that that would be too slow, and we needed a couple orders of magnitude. And one of the big breakthroughs is when we got to this molecule, which has 2 fluorine atoms next to the alkyne. And we engineered those fluorine atoms there because they have a perturbing effect on the energetics of the molecular orbitals that we thought would be beneficial. And we were right about that because just by putting the 2 fluorine atoms, we boosted the rate constant almost 2 orders of magnitude. So this molecule, which we called difo for the difluorocyclooctane, that became one of our most useful reagents for in vivo imaging, as I'll show you next. But just to complete the chart here, you could get another boost of reactivity by fusing 2 benzene rings in what I call the west coast and the east coast of the molecule. So this molecule was laboriously given the acronym BARAC by John Jewett, who made this molecule, who came to my lab in 2008 from Chicago. So this was on his mind. But now we're almost 3 orders of magnitude faster than where we started. And then you can get another kick by shrinking that 8-membered ring to a 7-membered ring. But you have to put a large atom in the ring. Otherwise, it's too reactive. So sulfur serves that purpose to kind of relax the strain a little bit. And this molecule was up to 4,000 times faster than where we started. So now that we had figured out how to create a faster bioorthogonal reaction, still using the azide as one component, we thought we were ready to go back in vivo. And by this point, we had become interested in using sugar imaging technologies to study changes in glycosylation in a more tractable model animal for optical imaging, which is the zebrafish. And what's great about zebrafish is they are translucent, so you can see right through them and image their body parts as they are alive, floating around in a dish. Also, they're a really nice model of vertebrate development because they can develop through in vitro fertilization all the way from the single cell embryo to a larval fish, which is kind of like a young adult fish, just in 5 days. And you see the entire developmental program unfold before your eyes, literally. So it's really great to work with if you want to do imaging platform development. So these are the kinds of experiments that Scott and Jeremy were doing back then, and this now takes us around 2007, 8, in that time frame. They would fertilize the embryos in a dish and then add one of our azido sugars to the media, bathing that embryo. And the cells in the fish would just take up these sugars from the media and metabolize them, and the azido sugars would end up on cell surface glycans on the different cells of the embryo. Then, in another step, they would add dipho linked to a fluorescent dye, like Alexaflor 488 is an example, and label all of those sugars with the fluorescent dye, and now you could image the sugars that were integrated into cell surface glycans during that pulse of treatment with this metabolic substrate. And then you could do this multiple times at different time points to label different kinds of sugars or sugars that were born at different time points during development. So what Scott and Jeremy would do would be to take their labeled embryo after one round of metabolic and chemical labeling, quench the unreacted azides, should there be any left over, through a Studinger reduction with tricarboxyethylphosphine, and then add more azido sugar to label a new round of cell surface glycans, either with a different sugar or the sugar at a different time point. And then you could distinguish the two populations of azido sugars with different color fluorescent dyes. So we would basically make fish with the different colors of the rainbow, where each color reflected either a different type of sugar or a sugar born at a different time. And that's what led to images like these. It's kind of hard to see in here because of the lighting, but I don't know, hopefully you can see. This is the head of a 5-day-old zebrafish that was labeled with 3 different sugars at different time points, and you can see the different colors are distributed in a very discrete way, which helped us to understand, the origins of different tissues in the embryo, where they came from and when. This is an earlier embryo. This is about 24 hours of development. And at these early time points, we could actually watch each cell on the surface of the embryo undergoing a round of mitosis, or cell division, which is what's happening as they differentiate and different tissues are forming. And these arrows are pointing to cells that are in the very late stage of a round of cell division. And if you blow up one of those and then take a time-lapse image of that actual mitotic event, you would see what's shown in these panels. So each panel here is the exact same frame. It's a mother cell, which is dividing to form 2 daughter cells. But we have different molecules labeled in the different frames. So on the far left here, there's a fluorescent dye on the DNA, on the chromosomes. So you can see the chromosomes are duplicating and then segregating during mitosis as the mother cell right now duplicates and forms 2 daughter cells. So that's what cell division looks like through the window of the DNA. And same exact thing is going on here, but now there's a different color dye, and it's on the membrane. So you can see the mother cell is surrounded by a membrane, and eventually the membrane fills in the junction of the 2 daughter cells as they form right there. There's a new membrane, and now you've got 2 separate cells. So that's what cell division looks like through the window of the membrane. But now we kind of added glycans to the list. Now we can monitor cell division through the window of the glycans. And you can see it starts out with kind of like a fuzzy coating around the mother cell, and it's fuzzy because the sugars are literally like hair all over the surface of the cell. But then late in mitosis, there is a concentration of these sugars at the junction between the 2 cells. They build up right there, and they don't actually fill in with the new membrane. They just stay right there at the junction. And it turns out that the sugars we were labeling here, which are on a certain class of proteoglycans, they are playing a role in segregating the 2 daughter cells, which was something that was invisible to the world before we started making these observations. And that sent us down a really interesting path of studying sugars during development. But as we were publishing these papers, I think it became clear in other sectors of chemical biology that this general platform of introducing a bioorthogonal or clickable functional group into a biomolecule and then using that as a handle to introduce imaging probes, biotin groups for enrichment studies, that would be quite useful far beyond glycoscience, which was our particular application of interest at that time. So other labs started publishing on related work where they would put the azide not into sugars, but, for example, into proteins, which, on this pie chart makes up more than half the mass of the cell, not counting water. And there's a number of different ways that groups have now put azides into proteins. For example, by introducing them into unnatural amino acids, which can be genetically encoded in proteins of interest or just used as metabolic substrates for temporally specific protein labeling. People have put azides into covalent enzyme inhibitors to basically study the activity of those enzymes in living systems or to look for on-target and off-targets of drugs of interest. People have put azides into nucleosides to image RNAs and DNAs, and people have put them into lipids to image lipids as they move around in the cell. So this concept is now, I think, sort of baked into the fabric as a platform technology for the life scientist, and you can buy azide or alkyne modified amino acids, sugars, lipids, probes, now from dozens of different reagent catalogs. Okay, so this is accessible to everybody. In recent years, I've become, like I said originally, more and more interested in the translational applications of bioorthogonal and click chemistries. And there's a number of them out there now. So I think the biopharma industry has already embraced these technologies for their own purposes. Some standout applications are, for example, use of these chemistries for making antibody-drug conjugates, which have gone hot and cold over the years since I've been in this business, but right now they're super hot, as a new kind of targeted therapeutic for oncology and beyond. And one of the applications of bioorthogonal chemistries in this space has been to make antibody-drug conjugates where the drugs are attached to the antibody in a site-specific way, which was not a capability that the industry had really until these chemistries came along. So now there's a number of different companies that have platform technologies for making site-specific antibody-drug conjugates using these chemistries. One of them is a company that I co-founded called Redwood Bioscience, which was acquired by another global company called Catalent. And I'll just show you what we do in that company. We developed a different bioorthogonal reaction that we can perform on proteins that we have pre-functionalized with aldehyde groups. And we developed a technology we call the Aldehyde Tag technology, wherein we simply introduce a 5 amino acid motif into a protein of interest just by cloning. So we clone in the sequence cysteine, X, proline, X, arginine, where the X's are variable residues of interest. And that motif is recognized by an enzyme called the formyl glycine generating enzyme, or FGE for short, which is naturally found in all of our human cells. And what that enzyme does is it oxidizes the cysteine to formyl glycine, which has an aldehyde group. So now there's an aldehyde on the protein at this motif that you genetically encode. So now what we do is we introduce that sequence into the sequence either of the heavy chain or the light chain of a monoclonal antibody, and we express that plasmid with the other chain of the antibody, sometimes with a little bit of extra FGE, so that the cells will just produce antibodies with aldehyde groups ready to go. Now we had to invent a chemistry for the aldehyde group that was bioorthogonal, and we came up with this thing we call the Hertz ligation. It's basically an adaptation of the Pictet-Spengler reaction. Anybody heard of that one? One organic chemist who memorized named reactions when they were in grad school, I'm guessing. So Pictet-Spengler chemistry is another 100-year-old German reaction. And our adaptation was to introduce this dimethylhydrazine group on the indole so that the product is a stable carbon-carbon bonded product, which was formed through an electrophilic aromatic substitution. Anyways, that chemistry allows us to attach linkers and drugs specifically to these aldehyde sites and nowhere else on the molecule. And if you're interested in the mechanism, one organic chemist over there, this is what happens. This hydrazine reacts with the aldehyde to form an iminium intermediate. which is then reacted through this intramolecular electrophilic aromatic substitution to form the product. And anyways, anyone here in the biopharma industry can avail themselves of this technology by simply reaching out to Catalent, because they will partner with you. And all you have to do is send them the sequence of the heavy and light chains of your antibody of interest, and they will make it with aldehydes and put drugs on it as you demand. So this is the bioorthogonal reaction that we have translated into a site-specific antibody-drug conjugate platform. But other companies have used this and other types of click chemistries to make ADCs. And in fact, I thought I would point out one of the FDA-approved antibody-drug conjugates, which is on the market today, which was originally developed by a company called Immunomedics, but then acquired by Gilead. This drug called Trodelvy, if you scan through the sequence here, you'll see there's the protein on the left, that's the antibody. This is the drug on the right. It's a toxic drug that kills the cancer cells that the antibody directs it to. But built right into the middle, you'll notice there's a triazole, and that triazole was a site where the chemists who manufacture this drug were able to conjugate two things together. And because, and probably with copper, I'm assuming this was a copper click chemistry reaction, fine in a test tube, fine in a manufacturing plant, you just can't do it in a living system. But this is now a drug that was enabled by click chemistry. Finally, I'll show you one other type of bioorthogonal chemistry that came from almost simultaneously from the labs of Joe Fox he's at University of Delaware, and Neil Devaraj, who developed this when he was a postdoc in Ralph Weislater's group at Harvard Med School, and they published these papers almost at the same time. This is a bioorthogonal reaction called the Tetrazine Ligation, and I'm a big fan of this chemistry. This is what happens when you take another strained cyclic system, but instead of an alkyne, it's an alkene. So this is trans-cyclooctene, and it's very reactive in an inverse demand Diels-Alder reaction, another ancient organic chemistry, with this molecule, which is a tetrazine. That's why it's called the Tetrazine Ligation. So these two things will react to form an adduct, which tautomerizes to form another adduct. This is a very fast reaction. It's a couple orders of magnitude faster than our fastest copper-free click chemistry. So that's why I love this so much. The other thing is you can do cool things with this reaction, like engineer it to release cargo. So Marc Robillard, who's a chemist in Europe, showed that if you introduce like a leaving group at this position, then when the trans-cyclooctene reacts with the tetrazine, you make an intermediate which is prone to eliminate that payload. So not only do these two things click together, but they spit something out. And that so-called click-to-release mechanism has been used by a number of groups as a pro-drugging strategy. And I thought I would mention the work of a company called Chasky, which is a Bay Area biotech company, because these are the first folks who've actually done a bioorthogonal reaction in human beings. And as a disclosure, I have been an advisor of this company for 8 years or so. And what they do is this. They have a mechanism to use click to release in order to have a drug become active only in a tumor microenvironment. And the way they do this is they make a hydrogel polymer that has been chemically functionalized with the tetrazine. And they will inject that hydrogel polymer directly into a tumor, like an intratumoral injection. And they've done this with a particular kind of bone cancer as their first indication. And the polymer just sits in that tumor, and it does nothing. It just sits there. And by the way, the polymer here is called hyaluronic acid. It's the same polymer that's used in as a cosmetic filler, right? So it's a totally safe material that's a natural polymer that is injected into people's faces, especially here in Southern California, all the time. But now it's got a tetrazine on it. So that polymer just sits there doing nothing. Meanwhile, they put the other reactant for this click to release, the trans-cyclooctene, they put that on a toxic drug in a place that causes the drug to be inactive. So it's a prodrug. And this is a chemotherapy called doxorubicin that's normally really toxic and has horrible side effects in patients. But when this modification is on it, inactivated. So they'll infuse that prodrug into the patient through an IV, and the prodrug is everywhere, doing nothing at all, totally inert. However, when the prodrug encounters that polymer, which is sitting in the tumor, the chemistry occurs in the human, causing release of the doxorubicin at a very high local concentration, which then kills the tumor without the horrible side effects normally experienced with this drug. So they just concluded a phase 1 study of this platform, gearing up for phase 2, and I'm really excited to see what they're doing with bioorthogonal chemistry in the human patient. So a big landmark at Chasky. But let me just leave you with this last slide because I introduced the concept of bioorthogonal chemistry. I showed you the origins of a few reactions like the Studinger Ligation, the copper-free click chemistry, and then our Pictet-Spengler ligation, and then finally this Tetrazine ligation from other labs. Those are the handful of bioorthogonal chemistries that are now in common practice in the world, but I'm of the mind that the future is very bright for the discovery of even more of these chemistries from that galaxy. So one of the challenges I like to put forth, especially in an audience that's rich with students, especially chemists, if you're thinking about ways that you could contribute as a chemist in the future, I would say inventing or discovering new bioorthogonal reactions. There's lots of open field to play there. So with that, let me thank my group, integrated over 25 years, which is impossible to do on a naming slide, so I'll just show you a couple of photographs. So this is a fairly recent rendition of my group at Stanford. You can see the sun is shining and it's warm and beautiful. And sorry, UCSF, but that's what we get down in Palo Alto. Wearing the Bertozzi shirts that we had printed just about a month before the Nobel Prize announcement. So it was great. We all got to go put our uniforms on for this picture. A few years earlier, during COVID, we had to do our group meetings outside with masks and 6 feet of separation, and the only way to film the group in that setting is with a drone. So I hear you had a drone battle competition yesterday, so it's fun, isn't it? So we rented a drone to take a picture. But after the Nobel Prize announcement, some of my alumni kind of pinged the whole network of alumni and organized a Zoom party to celebrate. And we managed to pull about 150 people together on very short notice from all kinds of different time zones, where we all did our best to have a cocktail so we could toast on Zoom. And so here's a lot of people from my group historically. But pretty much all the work I showed you here was done at UC Berkeley. So this really was a Berkeley story. And I owe them and that university a great debt of gratitude. So with that, thank you very much. And I think we can do our Q&A discussion. Thank you.
I was at Berkeley for 15 years. Where are they? Where's the other one?
There's some handheld mics, I think.
So, Carolyn, one question I had for you was, there's machine learning is having a big impact on protein science and protein engineering as a result of things like the Protein Data Bank, CASP, AlphaFold 2, David Baker's work on RF diffusion. Do you think that there's an opportunity for an AlphaFold 2-like moment for chemistry? And what would it take to enable that, given that a lot of the data sets are behind a paywall at ACS and Elsevier?
That's a really loaded question. So first of all, machine learning algorithms and techniques have already, certainly made their mark in chemistry in a number of different areas. Reaction development groups, especially catalysis groups, are benefiting now from these methods. Any area of chemistry where you generate a lot of data and make decisions based on empirical observations is ripe for disruption with artificial intelligence methods. And so already people are publishing some really nice papers where, based on a training set, you could predict what might be the best choice of a catalyst for a particular transformation of interest. That's already happening. Where there could be an enormous disruption is in the pharmaceutical industry, where you have decades of legacy data correlating structures with activities. But like you said, for the most part, those data are not mineable outside of those companies. And even within those companies, they're hard to mine because the data have different storage formats and they're not an easy way to sort of get them all in one place and searchable. But it's happening now. People have a mind for collecting data in a way that does make it useful for future efforts like this. Things that are so hard to predict, like when you make a molecule, what will be its oral bioavailability? It's totally empirical, and you can't figure that out until you go into animals, and it's very expensive at that point. So it's really hard to screen thousands of molecules in animals to look for bioavailability trends. It's just prohibitive. So if you could make better predictions based on machine learning, that would be a disruptive space. Is your molecule going to cross the blood-brain barrier? Is your molecule going to be metabolized in the liver at this rate or that rate? These things are hard to predict. They're all empirical, but there's tons of data. So I think machine learning algorithms could help make better predictions along those lines. Material scientists are really benefiting. So people who make composite materials where historically, you just blended things together at different ratios and screen them for a function, right? And some screens are cheaper than others, and so I think having predictive power could be useful there too. In glycoscience is a big data problem, generally speaking. The structures are complicated. The structural diversity of the glycome outstrips the proteome and the genome by orders of magnitude, and we have to make decisions based on sparse data. So I think there's probably opportunities there for disruption, but not much has happened yet. But I definitely feel compelled to hire undergrads in my lab who understand these things and can help us think about how we could leverage those techniques. Yeah. Oh. Sorry, there's a. You have to point. Yeah.
Oh, who's going first? I guess I'll go. I was wondering. So a lot like the three sort of characteristic reactions that you cited there all have their origins in like the 1960s. I was wondering, to what extent do you think that what is otherwise bioorthogonal can be mined from sort of like SciFinder-like databases, given what we know already, or priors on sort of whether the nucleophile, electrophile, or other participating group has additional reactivity to other moieties in vivo?
Yeah, it's a great question, and it's sobering to think about how much chemistry is in the literature that's not searchable using today's internet tools. So SciFinder is pretty good for anything from the 1980s or more recent, but below that you miss a lot, especially if there's no English translation. And the organic chemistry is a field that's old enough, right? That field started to mature in the early 1900s, right? It started in the 1800s, but in the early 1900s there was kind of a burst of big pivot, breakthrough kind of era there, and a lot of the really amazing basic discovery chemistries were done in Germany and in Japan, and they're not in English. And the structures, are not searchable. The graphics that were primitive graphics in those old papers, right? They were all made on printing presses. So all the molecules had to be printed with horizontal and vertical lines, and I mean, it was just, it's not ChemDraw, right? So the idea that knowledge is lost to the world because today's chemist doesn't know how to go to a library is a really scary thought to me. And I still tell my students, you do still have to actually literally pull books off shelves and flip through them to get to some of that knowledge. But there's so many gems of chemistry in that knowledge because that was a time when, especially in Germany, there was a lot of resources and funding for basic curiosity-driven chemistry. So people like, Hermann Staudinger didn't study azides and phosphines because he wanted to image glycans in mice, right? I mean, they were just curious about what's up with these reagents,
I have one follow-up question, which is very short. Okay, go to someone else.
Thank you so much for your talk. I'm curious, for bioorthogonal reactions in humans for real clinical applications, I imagine one of the major challenges is getting site specificity or cell type specificity for the delivery of the orthogonal compounds. So I'm curious, in addition to the hyaluronic acid example you gave, what are some methods that you're excited about or optimistic about for achieving more human
Yeah.
applications?
So that's a great, very insightful question. And I'm of the mind that what bioorthogonal chemistry could bring to drug discovery is better spatiotemporal control of drug action. And I have another company that I'm right now assembling, which is going to be directing bioorthogonal chemistries to cells in a cell type specific manner using, instead of these intratumoral injections with polymers, for example, using reagents that can be systemically delivered. And we have some ideas for how to basically allow drugs to assemble in situ, right, from fragments that might have better properties than the assembled drug has on its own. And there are examples of types of molecules that once they get to their target, they're amazingly effective, but they have a hard time getting to their target. They might not be orally absorbable, or they might not cross the blood-brain barrier, right? Problems like that, but their fragments can get places, right? So the idea of like using the fragments for the delivery part and then having them assemble into the drug once they get to the target space, I think is an interesting idea to explore.
So at the end, you showed these few bioorthogonal chemistries that have been described thus far, and I'm wondering if you can draw out, even just from these few examples, are there some principles that unify them? I imagine it's much more complicated than just using functional groups that are not found in biology. There must be a lot more, that can go wrong. So I'm wondering what's the universal aspect of this?
I mean, so we started out, the azide was such a standout functional group because it is so inert in biology, and yet it has more than one mode of reactivity. And yet in nature, in your body, it does next to nothing. And we knew that because, when I was sort of came of age during the AIDS crisis. So during my graduate work, I worked on HIV drugs and stuff. And AZT was one of the first approved, HIV drugs back in the late 80s, early 90s, which is azithromycin. And so people on AZT were taking grams of drug per day, and that azide went right through their system unmetabolized. So I knew the azide was a winner, even for human studies, right? Just because of that AZT and other drugs that have azides in them. And then after that, we started thinking about pi systems, so alkenes and alkynes, especially alkynes, because there are no alkynes in the human body that we know of. We do have alkenes, we have unsaturated fatty acids, but not alkynes. So that's where we started, but then later we started thinking more about, maybe we should be thinking about the periodic table more, because the elements in your body are pretty restricted, right? There's a lot of interesting elements on the periodic table that are not in your body. And for example, bismuth is one that I've always had a fascination with. Bismuth is not in your body. It's not an element people use much in organic chemistry, but it's actually quite well tolerated in the human body. And if you've ever sucked down a bottle of Pepto-Bismol, you are full of bismuth and you're fine, So that's another way to think of it is to kind of landscape the periodic table for interesting reactivities. Yeah.
Over here. Thanks. You were talking earlier about the importance of big data in moving various fields forward and how that intersects with machine learning. And I think one of the things that's been super important for nucleic acid chemistry is being able to read and write nucleic acids and being able to, get these massive sequencing data sets. I know, people have been talking about protein sequencing for a long time, and that's sort of a holy grail. Are we anywhere on being able to do this with glycans? Can we read and write glycans? What do you see as the leading technologies in maybe being able to do this?
Yeah, so that's a great question. And we are now much closer to reading them than we ever used to be because of advances in mass spectrometry. So the most powerful methods for sequencing glycans at the glycome level from complex mixed samples and stuff is by mass spec. That's another area where my lab, we have a project in what we call glycoproteomics, where we're trying to sequence the glycoproteome, not cutting the glycans off, but the whole glycoproteome holistically by mass spectrometry. You couldn't really do that 10 years ago. because you didn't have the resolution, you didn't have the sensitivities. And also we have better tools now for isotopic labeling, and machine learning is a big part of glycomics, because mass spec data sets are huge and complicated and full of data that is not searchable with sort of commercial software package. So that's been really transformative. The writing part is more complicated. There are enzymes that build glycans and enzymes that degrade them. And you can use those enzymes to create glycostructures by genetic engineering or in vitro biochemically. But it's much more difficult than just like reverse transcribing oligonucleotides. And in part, and also the structures have a lot more variety, and they're not based on a template, right? So you can't amplify them. So it's different and much more complicated. And I do have a mantra in my lab where I tell my students, and this has probably been your experience too, any problem that you can somehow convert to DNA sequencing, you win. Whatever the problem is, right? If it's sensing something or whatever, if somehow you can manipulate it so that at the end of the day you're just sequencing DNA, you'll always win. So if you could find a way to read the glycome by somehow translating it to DNA, you win. So think about how to do that. Yeah.
If there are no questions, I'll ask one. You've published on glycoRNAs.
Sorry, who's talking?
Hi.
Oh, sorry.
Sorry. You have a paper on glycoRNAs with Ryan Flynn. Have you followed up on any of that work?
Oh, yeah. So, do you know Ryan? Have you met him? Okay, so Ryan Flynn was a postdoc in my lab. He's now a professor at Harvard, and his labs are in, where is he? Is he in Boston Children's Hospital? Or no. He's in Children's, right? PCH? Yeah. He's at Boston Children's, but his appointment is in the stem cell department at Harvard. And when he was in my lab, so he's an MD PhD student who did his PhD with Howard Chang. And Howard is a nucleic acid biochemist extraordinaire. And so Ryan came to my lab with deep expertise in RNA biochemistry. And he came to my lab because he wondered whether there was an intersection between RNA biology and glycobiology. And it was a reasonable proposal at the time because, I won't go into the backstory, but there is a form of glycosylation that occurs in the cytosol nucleus, and it often happens on RNA binding proteins. So he wanted to study this relationship between RNA binding proteins, RNA, and this particular glycosylation motif. And then through pure serendipity, he discovered something completely unexpected, which is that certain small structured RNAs make their way into the secretory pathway in the ER and the Golgi, where they actually get glycosylated with the same kinds of structures everyone thought were restricted to proteins. And this was a mind-blowing discovery in many ways because there was no framework in cell biology in any textbook that would explain this phenomenon. There was no framework where you would understand how RNAs could get into the secretory pathway. That had not been described. And how RNAs and glycans could get chemically connected, there was no known mechanism for that either. So we put out a preprint on that because I barely believed it myself, and we were like, we need to get some feedback from them. And 2 years later, we published a paper on that. And Ryan took that finding with him to launch his own new lab at Harvard, where he has continued to work on that. He's got 2 papers under review right now. And most importantly, we now have figured out what we think is the chemical structure of the connection between the sugars and the RNA. So keep your eyes on Ryan Flynn, and you'll see all these papers coming out of his lab. Yeah, I'm pretty excited for what he's been showing. Yeah.
I think my question was kind of in line with David Palmer from earlier. I was wondering sort of to what extent do you think that based off of functional moieties, the space of bioorthogonal groups have already been explored to some extent, or like in the limit of like the diversity that you
I would like to think that we have just scratched the surface of what could be a much larger compendium of bioorthogonal chemistries. So, there's like a handful now that people use kind of, oh, there's a sign. I can't read, I don't have my glasses on. Okay. I see like someone's like this and I'm like, Hi. It's like your mother's calling. Oh, okay. But anyway, so yeah, I'd like to think that there's more to discover. I mean, we worked on this for the first, maybe 15 years when I was at Berkeley. And then we kind of transitioned into working on other things, and other labs have made contributions. And so right now my lab is kind of focused on other project areas, but I do think that there's a lot more to discover and develop. But there has to be a need and a motivation. So if everyone is satisfied with the 5 options on the table right now, then the motivation isn't there. But if someone's motivated.
I can ask you yet another question, or
I have a somewhat non-technical question. How do you manage and think about the divide between your foot in the academic world and your foot in startups?
Oh, good question. So I have really enjoyed working on projects where there's an opportunity to take it beyond just the publishing of the paper or the filing of the thesis, And the older I get, the more important this has become for me. So, in the early days, I mean, I can't, again, turning my mindset back to where I was back in the 90s, when I started my job. At that time, I really just wanted to do something that I thought was cool and useful. And when I said, in my mind, useful meant other chemists would use it in their labs and stuff. And even though everything always had a high level, potential impact in human health, it wasn't that important to me early on whether I was the one who tried to sort of realize that impact. Like, it was enough for me just to publish papers and then let other people use it as they want, But then, you get older, right, and you start to see potential impact, and you realize that the runway, especially in biomedical science, right, the runway between the discovery and the impact on patients can be, is decades sometimes, if ever, right? And so the older you get, you run out of decade-old units, right, of time. So, right now, like, if I have an idea today, and I hope that it impacts patients, if I don't, put in the energy to try to make that happen, it might never happen, and there's not much I can do about it. Because 20 years from now, if I'm still alive, right, I'll probably be retired, and who knows, right? So, so the older I got, the more important that became. So when I was at Berkeley for 19 years, I started one company. That was Redwood Bioscience, the Catalent company, the Aldehyde Tag, so the ADC company. I moved to Stanford in 2015, and I've started 11 companies in 8 years. So that's, because I'm starting to feel like, oh my God, every year I get older, And the other thing I've found is students now seem, on average, more interested in not just filing their thesis and walking away from the science, but taking it and being a founder of their own company with me, So I even have students who, they meet with me, they're thinking about what lab to join, and they say, I'm interested in the science that you do, but if I join your lab, I want to work on a project where there might be a chance for me to start a company with it down the line. Like, they're thinking about that before they even choose their thesis project. Not all of them, but it happens, right? And that's kind of a more recent phenomenon. So I think just the culture, the ambition, the career vision of students today is more favorable towards entrepreneurship than it was in the 90s, And my experience is a manifestation of that. I'm delighted when I have a student who's like, I'm so pumped up about this. Now that I've published these papers, I want to translate this. Let's form a company, and I can be helpful. Is that your experience too as students?
Thank you so much.
Thank you.